Hello everyone and welcome to our video on muscle contraction. This is part of our Module 5 Part 5 series looking at those plant and animal responses, and in this video we're going to be having a look at specification reference 5.1.5 L Part 1, the structure of mammalian muscle and the mechanism of muscular contraction. So first thing we need to do then is understand the structure of muscles. Now, what we're going to start with are these things called myofibrils. If we break that word down, wherever we see this word myo, that relates to the muscle. So wherever you see myo, it's always something to do with muscle there. So myofibrils are actually the bits that are going to be contracting within the muscle. So they're the contractile elements, the bits that will undergo contraction. And these are made up of two types of protein filament. So we've got our thick filaments made of a substance called myosin, and that's sometimes referred to as the dark band. And then we've got the thin filaments made of actin, which is the light band. Now, what we find is those thin actin filaments are held together by a structure called the Z line. So if we have a little look at the diagram in the bottom left first of all, this is a typical diagram of one of these myofibrils. So what we've got, the red lines that we can see running down here, these are going to be the thick filaments, the myosin filaments. And then where you have a little look a bit further along, you've got this light blue colour thinner filaments, and they are the thin ones, made of actin. Now, where I've just put that arrow, you can see there's this little zigzag line that runs down, and that is our Z line all the way down there. If we have a look at what this is under a light microscope, then that gives us this diagram on the right. So what we've got, our Z line is going to be this dark line, very narrow dark line that we've got there. Then we've got these two lighter sections either side. So we've got these light sections here, and that makes up the I band. And then we come into the darker little bits as well. So if we have a look in the center there, we can see we've got these darker shaded ends with a lighter grey area in the middle. And what we've got is the A band stretching right the way across there. And the little bit in the middle here is the H zone. So what we're going to need to do is get our bands, zones and lines all correct in our heads. If you're mixing up the letters and the band, the zone or the line, you're going to lose marks on this. So make sure you do know what's a band, what's a line, what's a zone, and where they are both on diagram form and on those light microscope images. So that H zone that we've just talked about then is the bit that contains only myosin. So that only has the thick filaments. The I band is the region that's got only actin, so only the thin filaments there. The A band is going to be the total length of the thick filament, and therefore we're going to see both actin and myosin being present. And the reason for that is that if you consider the full length of that thick filament, so we've got our thick filament running right the way across, then our thin filaments actually overlap to an extent. So we've got our actin, the thin ones, overlapping with the thick filament of the myosin. You will also see that there's a region called a sarcomere, which quite simply is just the distance between those two Z lines. So you can see it on the diagram beneath there. We've got one Z line here, one Z line here, and between those is a single sarcomere. If we now have a look in a bit more detail at the structure of our thin filament. So the thin filament itself has two actin subunits twisted together. And if we look at the little diagram on the right, we can see those as the kind of white circles. So what we can see if I use a little colour, so we'll have a green one, so that one's running across this way. And then if we have our little blue line, you can see there's a second one running that way. So we've got these two actin subunits twisted together. What we then have is that dark red browny coloured bit is a substance called tropomyosin. and that's wrapped around the actin subunits. And attached onto the tropomyosin, we've got those green little blobs, which are troponin. And troponin actually has 3 polypeptides: actin, tropomyosin, and calcium binding points. So what we find is that we've actually got on that troponin these 3 different places it can bind: onto actin, onto the tropomyosin, and then onto calcium ions. So that's our thin filament. We've got the tropomyosin, the actin, and the troponin all twisted together in this very clear structure. The thick filament is made up of the myosin filaments. And myosin's a little bit special in the fact that it's actually got this little structure that's got 2 heads that stick out from the end. So if we have a little look, you can see it here, that we've got our little myosin, and then we've got these 2 heads sticking off. And they've got different binding sites. So the green one on our diagram is an actin binding site, and the yellow coloured one is the myosin ATPase binding site. And you can see how they arrange themselves in that second diagram there, where you've got the head sticking off on one side and the other side. So that makes up our thick filament there. So we now need to understand the process by which our muscles will contract, and this is referred to as the sliding filament theory. And basically this is just looking to see how those thick and thin filaments slide past each other, and when they do that, it shortens those myofibrils. Because if you think about moving, and you do it with your fingers to be fair, if you slide your fingers over each other, then the distance from one palm to the other gets less. Same idea here. And if we have a look, we've got these 2 diagrams to show us what's happening. So relaxed is at the top, so that is our relaxed state, and then the bottom one is our contracted state. Now what we can actually see in the relaxed state is that we have this wider eye band. So if you look at the eye band, which is this region where it's only the actin, then we obviously have a reasonable width there. But when we actually contract, you can see it's absolutely tiny, okay? We don't really have much of an eye band left at all. So what we see is in that process of contraction, the eye band gets much smaller. The next thing that we will see is that those Z lines are obviously moving closer together. So we can see we have a Z line here and a Z line here at the top, and then they are moved closer together. So the Z lines move closer together. We then have a greater degree of overlap between our thick and our thin filaments. So the myosin and the actin have a greater degree of overlap, which we can see in the diagram with the red and the blue lines. But please be careful. The A band, which is our thick filament, that doesn't change size. Because it's obviously spanning the full distance of that thick filament, the thick filament isn't shrinking. Therefore, your A band remains the same width. So just go careful with that when you're writing an answer to do with these differences, that A band will remain the same size. It's the I band that gets smaller. So how does it actually happen? What happens to make a muscle contract? Well, first thing, we need to understand what communication is happening between our neuronal system and the muscle itself. So when that nerve impulse arrives at the muscle, we're arriving at what we term the neuromuscular junction. So neuro because it's a nerve, muscular because it's connecting to a muscle. So neuromuscular junction, NMJ, this is talking about the connection between our neurons and the muscles. So when that nerve impulse arrives at the muscle, we're going to see a depolarization of the sarcolemma. And the sarcolemma is basically the membrane of our muscle there. As a result of that depolarization, we then have a transmission throughout the fibre down these structures called the T-tubules. And you can see one of the T-tubules just here. So basically what we see is as that neuromuscular junction transmits that impulse across the synapse through the use of vesicles containing a neurotransmitter, we depolarize our sarcolemma and that travels down those T tubules. And what happens is, as that depolarization travels down through that muscle fiber, then we open the calcium ion channels. And those calcium ions will be released from the sarcoplasmic reticulum. And sarcoplasmic reticulum is kind of like the endoplasmic reticulum, just in muscles. So once we've got those calcium ions, they're going to diffuse amongst the myofibrils. And remember what we said earlier, that troponin has a binding site for calcium ions. So the calcium ions bind to the troponin, and when they do that, it changes the shape of the molecule. And when it does that, it's going to pull the tropomyosin, that kind of reddy brown line that we can see on the diagram there, away from the myosin binding sites. So what that means is the myosin heads can then bind to the newly exposed myosin binding sites. So now we formed a cross-link between our myosin and our actin. What we then find is ADP and inorganic phosphate that's in those myosin heads will be released, and that means the myosin heads bend. Now, if you kind of imagine that we've got our little head facing this way first of all, then what happens is it's going to bend, so it's going to move inwards. And because that's bound onto our actin, it pulls it inwards. And this is something we refer to as the power stroke. So just by changing the shape of that myosin head, it's dragging that actin filament closer to the centre of the sarcomere. What we then find, ATP will bind to the myosin head, and as it does so, it breaks those crosslinks and it releases the actin from the myosin. Now the myosin head acts as an ATPase, hydrolyzes the ATP into ADP and inorganic phosphate, and therefore the head returns to its original position, and that brings it conveniently aligned to the next myosin binding site on the actin. And basically it repeats. So we keep kind of ratcheting the actin further in. Each time we're binding, obviously the myosin to the actin, we get that change in shape and it pulls the actin a bit further in. ATP binds to the myosin, the crosslinks are broken, it returns to its original place and continues. As a result of that happening over and over again, the muscle contracts. So here's our little summary diagram just showing us how it all works, and this is a good thing to basically print off and check you can explain each stage of our diagram. So we'll start at the top here, okay? What we've got then, our myosin head is able to actually bind to the actin and it forms that cross-link between the two. What we then find is that the ADP and inorganic phosphate get released, which leads to that change in the shape of the myosin head, and therefore it drags the actin filament towards that centre, the M line. ATP then binds to the myosin, which results in the release of the myosin from the actin, so we've broken that cross-link, and we hydrolyze the ATP into ADP and inorganic phosphate, which then returns it to its original position, and therefore the myosin head is now lined up with the next actin binding site, which means the process repeats. It is worth remembering that we've got to have this ATP present to break those crosslinks or cross bridges between the actin and the myosin. If we don't have ATP present, then we can't break them. So basically the actin and the myosin remain attached, those cross bridges are still there, and because of that the muscle remains contracted. So this is what we actually see happening in something like rigor mortis, which occurs after death, is that we're not making any more ATP, and therefore what we find is there comes a point where there's not the ATP to bind to obviously our myosin. As a result, the actin and the myosin remain bound, and therefore the muscles are in a contracted state, hence that stiffness that you get in a dead body for a period of time anyway. So where does this ATP come from then? Well, muscle cells actually have a lot of mitochondria because quite clearly they need lots of ATP for this contraction process to happen. So we're seeing a link back to our module 2 work on those subcellular structures. The mitochondria, obviously the site of aerobic respiration, therefore where the ATP is generated. Now, quite frequently, that's going to be sufficient to meet the needs of our muscle. But there are, of course, instances when that's not going to be enough. Think strenuous exercise. So what we've got are three mechanisms which are all designed to help us meet that additional ATP need. First one is having this specialized type of hemoglobin called myoglobin. So myo obviously referring to muscles, and globin obviously the bits that carry the oxygen. And what we find is this myoglobin that's found in muscles will release oxygen when that concentration is very low. So if you think about the situation, you've been doing some strenuous exercise, therefore the amount of oxygen you've got circulating within your body is starting to be reduced, then we're going to need a backup plan, and that's our myoglobin. So the oxygen concentration is starting to drop, myoglobin will release the oxygen and therefore gives us oxygen for aerobic respiration to continue. And obviously aerobic respiration produces like 38 ATP molecules for each molecule of glucose respired. Second way that we can deal with this is using anaerobic respiration for a short period. So anaerobic respiration, remember, is only utilizing the glycolysis part of respiration, and therefore we only have a net gain of 2 ATP molecules. The downside, we're also producing lactate, and obviously that's the problem, because the lactate, when it accumulates, causes that fatigue, and therefore we're not going to be able to sustain this anaerobic pathway for long. It's merely a few seconds. The final option available is the ATP creatinine phosphate system. Now, the ATP creatinine phosphate system is providing a little bit of extra ATP for a short period. So in our muscles, we've actually got this stuff, creatinine phosphate, or PCR for short. And what we're going to find is that's able to provide phosphate to convert ADP into ATP. So what we're really seeing is our ADP, which is present within those cells, is then going to use the phosphate from the creatinine phosphate, PCR, to then form ATP and obviously the creatine. Now that's an extra few seconds of contraction. And what we're really seeing here is not something that's going to give us endless amounts of ATP. It's just that tiny bit more that our body might need just to escape from danger, for example. So make sure you know those three different ways that our body gets the ATP required for muscle contraction. As always, I hope you found this video useful, and subscribe to the channel to get the latest updates on when the next videos are uploaded. And of course, head on over to the website where you can find a range of other resources to help you in your A-level biology studies.